Yeah, so I'm going to sort of talk about guitar strings and balloons a little bit, and I'll explain why, hopefully by the end. And before I do anything else, I want to highlight the people who did the work, including the folks in my group. I'll highlight some work done in collaboration with Christina, who's here, and Jen Schwartz, Shane Bell, who I don't think is here today, was here yesterday, Max B, who's in Boston, and who's absolutely fantastic. He's at Northeastern, tenure track there. And the work I'll be highlighting today is largely with Brian Feig and Carson at Delft. Okay, so if I want to understand the final morphological shape of an object, which is what a lot of us are trying to do, then it depends on the forces that act upon it, whether those are externally generated or internally generated by cells, and the properties of the material, or its rheology. And so when I sort of first started thinking about these things, I was really surprised, I think, that the emergent rheological properties of groups of cells, I don't think was necessarily extremely well understood. The properties of single cells, there were a lot of techniques for that, but maybe not this. And the other point that I want to make is that I'm not going to be talking very much about genes today, but the reason I think this sort of way of thinking about things is useful is that genes control both of these things in cells, both the forces that are generated and sort of the mechanical properties of the material. And so I think by organizing things into these buckets, like the what genes affect the rheology of the material, what genes affect the forces, that can lead to sort of a way of thinking about self-organization in patterns that has a smaller sort of subset of parameters. Okay. And so I sort of want to argue that the rheology, the large-scale mechanical properties of tissues, are important for biological function. And this is a recent paper out of Oche Campos' lab. Dr. Becker is here somewhere. And so the idea is that there is a change in the mechanical properties of the tissue in the zebrafish tailbud. So we saw some talks about somite formation. These are the somites forming. This is the tailbud region. And so sort of the idea is that this material is much more solid-like than this material, and they argue in this paper that that's actually, that gradient in sort of material properties is really important for generating the final shape, this elongated shape that you see in the zebrafish tailbud. So what I'm going to focus on today is rigidity transitions. And so I'm going to be asking the question, what do cells have to do in order to generate, say, gradients in material properties like this? And the idea that I want to leave you with is sort of that in certain parts of development, you need cells to either undergo really large displacements, they move over long distances to get to where they need to be, or the entire tissue deforms globally by a very large amount. And that you would expect to be associated with a fluid-like rheology or behavior of the tissue. And then in other times in development, say when you have buckling or you need to support shear stresses, then you need a solid-like rheology. And so it's maybe not surprising to think that this is an organizing principle that organisms could take advantage of to sort of program in different behaviors during development when you need them. So there's experimental evidence that confluent biological tissues change their behavior as a function of cell shape. So by confluent in this talk, I'll just mean cells that are not overlapping one another and they don't have gaps between them. So they're sort of filling all of space. And so this is a sort of experiment that's in Jeff Fredberg's group. It's a really beautiful setup where they take cells from the lungs of human patients and they culture them at an air-liquid interface. And so you're looking at a monolayer of cells at an air-liquid interface. And now these are sort of bird's eye view. So you're looking down at this monolayer and you're measuring how fast the cells are moving in various spots. And you see that, if you've cultured the system after 3 days in culture, the cells are sort of changing neighbors and displacing a lot. This is the speed measured over some time scale, so they're moving a lot. By day 6, they're hardly moving at all, and by day 8, they're really not moving. And you can do something similar in systems. So these are breast cancer epithelial cell lines, so they're not primary cells like Zeb was talking about, but nevertheless, you can study them. This is sort of the standard. So these are MCF10A cells. And so if you sort of look, and these are sort of 436 cells that are sort of a model for malignant cell type, and you sort of, each one of these things is a ball of thousands of cells. So these are thousands of cells, and you're just bright field imaging them in a petri dish. And then I'm going to show you what happens over a time scale of a day or so, is that you can see that in this case, the cells sort of, these groups of cells partially fuse together, but then they get stuck. And they arrest in this dumbbell shape. And at the very end, it's getting clearly unhappy, but you can look for cell death markers and things like this, and sort of, there's not much happening over the first day, other than these things are not rounding up as you would expect 2 balls of fluid to do, right? And so this is sort of when you robustly get this shape, and it actually rests in nearly exactly the same dumbbell shape every time. And so that really suggests that the rheology of these balls of cells is solid-like on some level. This couldn't happen unless they were solid-like. Okay, and then you can look at these tumor cells, and what you find is the cells pretty rapidly fuse, these balls of cells fuse together and round up, like you're seeing oil, like you would see in oil droplets or in water. It's very similar. Okay, and so what I'm trying to highlight here is this is experimental evidence also of a sort of fluid-solid transition in the system. Okay, so the model that I'll be discussing today was already introduced by Christina Marchetti yesterday, but I'll go over it again briefly. I'm going to discuss it in terms of looking at a bird's eye view of a two-dimensional epithelial layer. And if you look at a layer like that and cell membranes are labeled, you can see that each cell has an area, a cross-sectional area A, and a cross-sectional perimeter P. And lots of folks have sort of over time developed basically a very similar set of models for this. And the idea is simply, if you're a physicist, what this is it says there's a homeostatic area that you want to maintain, and you pay a quadratic penalty for being away from that. And there's a perimeter that you want to maintain, and the lowest Taylor expansion is a quadratic penalty away from that. If you're a biologist, then you can actually go in and do pipette aspiration experiments and measure interfacial tensions. You can also look for evidence of actin-myosin contractility that may drive these higher order terms. This term comes about from bulk 3D and compressibility of cells. So the idea here is either you can view it as a minimal model or as something with experimental evidence, but a lot of people have used it successfully. I want to highlight in particular sort of, you can non-dimensionalize the equation, and there's sort of one really key parameter, which I'll call a target shape index. It's the target perimeter, the perimeter a cell wants to have, divided by the square root of the target area, so that's dimensionless. And the idea is as follows. If you have cells that are confluent, they can sort of have a large perimeter relative to their area, so they sort of look isotropic like this, okay, and that will be a large value of this target shape index. Or the best they can do in sort of a confluent tissue is look sort of almost hexagonal, and so they're very isotropic, and in that case, this has a lower shape index, lower perimeter to area ratio. Okay, and so we've spent some time over the past few years trying to understand basically the mechanical properties of this model. And so we sort of wanted, there's actually some really beautiful work, I want to highlight it, by Frank Hiller's group looking at the ground states of this model here, and then this paper is also really beautiful. And we were interested in the metastable states because that's what you often see in experiments, and in particular we were interested in transitions between metastable states. So this is a video of a simulation where we've just had this one cell execute basically a series of cell rearrangements. So this is just like a single cell perturbation, but it looks like it's migrating. It's not, that's just a calculation we're doing to calculate energy barriers between states, actually. Once you try to calculate the energy barriers between states, so this is the energy barriers between two states where the cell has changed neighbors, and this is this target shape index. And so you can see that if the target shape index is below something like 3.8, then there's a finite energy barrier to rearrangements. and if it's greater than 3.8, that there's essentially no barrier to rearrangements. And so the idea is that in this system, which is confluent, so it's not changing its packing fraction, you can get a change from fluid-like to solid-like behavior just by changing cell shape. And if you're a physicist, you can actually try to get a good handle on where the transition point is by looking for a scaling collapse, and we did that, and you get a really beautiful scaling collapse over orders of magnitude. There's some things here that I won't talk about. Okay, and then the work that Christina sort of discussed in a little bit more detail yesterday, you can see that you can also then say, okay, but in real life, that was a sort of like a zero temperature calculation. In real life, cells can move around and there's fluctuations, and so what happens to this model in the presence of fluctuations? And so you can see that you can get sort of this transition between fluid-like and solid-like behavior, also by increasing the sort of motile forces that cells can exert. That will tend to fluidize the system, and as Christina pointed out yesterday, if you increase the persistence with which cells move, that will fluidize the system. Okay, and so I think the sort of most useful thing for people who do experiments, and I was really surprised by this actually when we realized it, is that if you look at a snapshot of this simulation and that simulation, just so don't try not to pay attention to the dynamics. Look at a snapshot, and you can tell the cells look different, right? They look different. These ones look more anisotropic than these ones, right? Their shapes are different. And so what we realized is that instead of, because the target shape, the shape a cell wants to have is some nebulous model parameter. But a very easy observable is what's the shape a cell actually has, okay? And so you can calculate that. And what we find in the simulations is that basically in the fluid phase, this observable changes, and it changes. So basically in the fluid phase, you can have exactly the shape you want, and so it changes in the fluid phase, and it gets the shape gets sort of more and more elongated as you get more and more fluid-like. Whereas in the solid, you're basically pinned at a number, so you're sort of pinned close to 3.81, and that's sort of true independent of any other model parameters. And so that's kind of cool because that's a prediction for experiments. It basically suggests in these movies, let me show you these movies one more time, if I can. So this is sort of a movie now of what happens on day 3, and you can see cells are moving a lot. Movie at day 6 and movie at day 10. So these are really jammed, and you can sort of say, okay, I will measure the cell shapes for each of these pictures, and what I expect is that on day 10, I expected this system to have this sort of cell shape, which is basically 3.81, which is this sort of number that's predicted a priori from the theory. And sort of, sure enough, that is what you see. So sort of this is this cell shape that you measure, and it sort of rises away as you go into the fluid phase. So that's kind of cool. And then the sort of other... Yes?
Just a quick question. If you were to relax into solid phase, but not from a fluid, but say by having cells divide to create a fluid, would you also expect to reach the same state, or could you end up at lower at different values of P?
So that's a really great question, and there's 2 answers. The first is that there is an ordered ground state to these types of models, and if you have a preparation protocol, which instead of cell division is shear, then you very easily find that ground state, and actually can get to a lower value. You can get to the ordered ground state. If you have cell divisions, actually, it turns out that you sort of generically end up in a state that sort of, there's a subtlety, which is that over long enough time scales, cell divisions will fluidize the system, but on intermediate time scales, the solid will still look like this, and it will still have the rheology of the solid with cell divisions. There's one additional wrinkle, which is that in like MDC monolayers, or something which really have cell divisions, there's also this feature that we know that sort of contact inhibition of locomotion means that there's, when cells are sort of dividing and dividing more and more, that there's sort of a downregulation of cell motility that goes with it. And so there's actually something we could talk about offline where you may get a different cell shape through cell divisions because you've sort of turned off the temperature in a weird way. You've turned off the motile forces in a weird way, and that's a different state. Okay.
Okay, so the other thing I wanted to mention is that we have focused a lot of our efforts on thinking about cellularized tissues, where the cells are confluent and all the cells are touching, but a lot of developmental systems is also made up of extracellular matrix, right? Acellularized extracellular matrix, or some with a few cells in it. And the thing that sort of struck us is that there's been a lot of work recently showing that these biopolymer networks also undergo a rigidity transition, but now it's as a function of shear strain. So you take a chunk of this material and you shear it, and then it will rigidify. It sort of has a very low modulus, so it's a very weak solid down here, and then sort of at a critical strain, it will basically have a kind of a fairly sharp turn on of a much higher modulus. So this is a logarithmic scale, so this is several orders of magnitude. So it basically goes from being extremely floppy to much stiffer at a characteristic strain. Okay. And then, the reason why I bring this up is because actually if you look at the mathematics behind models for these vertex models and these biopolymer networks, the structure is actually remarkably similar. So I explained this one to you already. This is the non-dimensionalized vertex model. And what I'm showing you is one of the standard sort of spring network models for biopolymer networks, where you have a penalty for changing your angle, and that has a modulus. And then you also have a penalty for changing the rest length of your spring or your fiber, and that has another modulus. So there's a rest length, and you change away from it. Okay, and so to simplify things for the most part in my discussion today, I want to point out that actually you can get rid of these terms and recapitulate a lot of the same behavior in these models. So I'm actually going to forget about this term in large part and this term in large part. And then these two look really, really similar then, obviously. Okay, so the models are very similar. So the question that we're going to be asking is, do these observed rigidity transitions have anything in common? I'm going to also sort of ask, because it was brought up yesterday, sort of changing the number density, right? Because in jammed systems, you change the number density, and that's a way to sort of get rigidity. And so that's pretty well studied, and I'd like to know whether these rigidity transitions have, are they sort of the same story as those which we understand very well? Very is a strong word. Somewhat well. Okay, so Maxwell, so when you're thinking about rigidity, sort of Maxwell was one of the first people to think about this, and basically there's this constraint counting argument that if you look at the number of degrees of freedom in your system, in this frame there are 8 degrees of freedom, 4 points, and in 2D each has 2 ways it can move. And then there are 4 constraints. Each bar represents a constraint. And then there's rigid body motions that are possible. There's 3 of those in 2 dimensions. And that leaves one non-trivial floppy mode. If I sort of say, okay, I take the number of degrees of freedom, I subtract off the constraints and the rigid body motion. Okay, and so here I'm showing you that mode. It's like a shearing mode of that frame. And so this system is what would be called under constraint, because it's, the number of degrees of freedom is larger than the number of constraints. Okay, of course I can introduce an extra constraint, and that will rigidify my system, and I find that yes, in fact, it does. Okay, and this Maxwell constraint counting, I would say, is sort of surprisingly good at explaining rigidity in jammed particle systems. So our intuition is that materials solidify as I sort of increase the density of the system, right? And in disordered systems, it's actually not completely trivial how that works. So in disordered spheres at 0 temperature, which is called particulate jamming, the way it works is that there are n times d degrees of freedom, and then these particles are sort of squishy spheres or hard spheres, and they make constraints with other particles. And so there are n times z divided by 2 constraints. So z here is the number of contacts per particle, and I divide by 2 because the particles share a contact.
Same radius?
What?
Same.
Yeah, this is a same radius monodisperse. This is the simplest thing. Okay. In mean field, so actually this argument works for polydispersed spheres, but it doesn't work for ellipses and things with friction. Okay. In mean field, that means you sort of say, okay, well let me just say that I expect that everything is the same, and so then I should expect rigidity to occur when I set this equal to that, and that gives me that I expect that to happen at a coordination number that's 2 times the number of dimensions. Okay, and at low densities, the number of contacts I have is less than this, critical point, and the system is under constrained. And then at high densities, the coordination is greater than that critical point, and the system is over constrained. Cool. Okay, so this is sort of why disordered solids solidify in a nutshell. Okay. Now, I'll mention that this actually means, if you think about it, that these systems are really highly self-organized in a special way so that this mean field counting works. Also, you can basically design things that have these topologically protected excitations, because this is basically a rank nullity theorem, and so there's a lot of cool stuff. Okay, cool. Okay, what I'm going to tell you is that polymer networks and these vertex models are in a different class. They're different from that. They don't do that. Okay, and so to explain that, I have to explain states of self-stress. Okay, and the idea is that this thing is a rigid box, because when I count the number of constraints and the number of degrees of freedom. But if instead of putting my next bar here, instead I put an extra bar in this frame, then my system would still be floppy, even though in mean field constraint counting I would expect it to be rigid. And I can take that into account by saying, well, actually there's a state of self-stress that comes in. And states of self-stress are where the displacements are basically states of stress in the system that don't actually end up in displacements of any of the nodes. Okay, so why am I telling you this? Because this gives an alternate path to rigidity in under-constrained systems. So if I have a system that's under-constrained by constraint counting, if the geometry of the system induces a self-stress, so if I have some sort of external thing that I'll explain examples of, the system can rigidify even if the number of degrees of freedom are larger than the number of constraints. So let me give you some examples, because I know that's a little bit, abstract. Okay, so a guitar string is a good example, and I'm going to represent a guitar string as n segments, each with a rest length L0. Okay. And so there's a geometrically compatible state where all the springs are happy. They can all attain their rest length. And that's in the case when the endpoints of the string are such that the number of segments times the rest length of each segment, right, is greater than the distance between the two endpoints. So this is a compatible. Okay? There's a geometrically incompatible state where no springs are happy. Okay? They're all stretched past their rest length, and that's when I stretch the box beyond the rest lengths of the individual things, okay? And the rigidity transition occurs when the total length, L, this total length L divided by the number of segments is equal to L naught, and we'll call that special point L naught star. This is the transition point. And I want to note right away that you can achieve this point either by thinking about changing the size of your box, right? Or equivalently by changing the rest lengths of my spring. So if I kept the box fixed, and change my spring rest lengths, I could achieve this, or vice versa. Good. Okay, so I'm going to represent this in a different way, which is, I'm just going to show you sort of, this is the spring, this is a representation of the string length, okay, and I'm changing the string length, and it's floppy if the string length is long, and then there's this critical point that I hit where I sort of rigidify the system, and then when I keep decreasing the string, then actually this is all of the, everything that's beautiful about music, because then I can start hearing different pitches because the tension in the string is changing. So a lot of what I'm interested in is actually the behavior of the system right past this critical point. Right below this critical point is really interesting. And so that's what we're going to be talking about today. Okay, there's another example. I like this example, balloon, okay? So a balloon is floppy, okay? And you blow it up and there's sort of a critical point at which you put just enough air into the balloon that the material that makes up the balloon encapsulates that air without stretching. Right? So I have to put in enough air to make that balloon sort of just start to become taut. Okay? And it's exactly the same thing. There's a critical surface area at which that balloon becomes taut because I've basically made it geometrically incompatible without stretching. Okay? Cool. Okay, so this is the surface area of the sphere. Okay. So then I want to know what happens in disordered systems in dimensions greater than or equal to 2 where it's not so simple. In the guitar string, I could just look at a picture and figure out what this was, right? In the balloon, actually, it's about this critical point, this is roughly a sphere, so the critical point is when the surface area of the balloon encapsulates a volume of the sphere. right? So I can figure out that critical point. What happens in these disordered geometries? And so you can do simulations like this of spring networks where you're sort of doing pure shear. So I'm showing you a little bit of pure shear in this network. And you can see the colors are telling you the tensions in the network. And then at a certain point, the sort of the system sort of there, it pops in. There sort of begins to be tension in the network. Okay, so we want a general way to think about the geometry of materials close to these types of transitions. Okay, so I already gave you this example. So just to fix terminology, let L naught star be the average spring length at the rigidity transition. And in the floppy phase, all the springs are able to achieve their rest lengths. So what happens to the spring lengths, the observed spring lengths, when I sort of go below this critical point? Okay, and what I want to sort of, I'm going to tell you what I'm going to tell you, which is that if I go below L star, then we sort of have this claim that the geometry of the system follows a simple and robust scaling form. So if I look at fluctuations in either the shape or the spring length, then I can basically say that the sort of geometry of the system as I go into the rigid phase has a linear scaling with the standard deviation of the fluctuations in the length. And I'll sort of explain why that's interesting, because the scaling relation allows us to unify a whole bunch of existing predictions that have sort of been generated in nice, but maybe not in a unified way. And it also explains the behavior of these vertex models for biological tissues and shows that they're actually the same. And then it allows us to make new predictions. Okay, so let me just walk through briefly an example. So I showed you the energy functional for a spring network. Basically looks like the spring minus its rest length to the second power. Okay, and what I want to emphasize is that can be transformed into one term that's quadratic in the mean minus so this is the observed mean length of the springs minus their rest length. And then these are the fluctuations. Okay, this is just algebra. Okay. So what this means, though, if you think about it, is the system needs to compromise because it can't be happy. So it needs to compromise. It can't get its rest length. So it needs to compromise between minimizing this quantity and that quantity because they're both quadratic. Okay. So this minimal length function describes the minimum average length possible in the rigid phase. And so I'm not going to go through the math, but because close to the transition, there's basically one state of self-stress that enters at the rigidity transition. Using compatibility matrix and a few other tricks, you can show analytically that this gives rise to a linear scaling. So this is an analytic result that I'm not going to go through, but I'm happy to tell you about it. Okay, so this is an analytic result. And so before I go on, I want to sort of, in this case of the spring networks, I want to emphasize one more time just so you get the picture. What we're doing here is we're either expanding our box, and that's exactly equivalent in the model to changing the rest length. So I'll show you two movies. So this is changing the box, and you can see that when I change the I isotropically change the box, there's a certain point where the tension comes in. And this movie over here, I'm not changing the box, instead I'm changing the rest lengths of all the springs uniformly, and you get exactly the same result. I'm just trying to give you a visual for that. Okay, and so when we do that, then you can say, okay, does this analytic prediction work in numerics when I do those types of numerical experiments? And basically this is a plot showing you that yes, it works really well, and moreover, we can actually explain how this coefficient here scales with the coordination of that network. So in collagen matrices and other sort of constituted in vitro systems, they can control the coordination of those networks, and so this is sort of explaining how you expect the scaling to change with that coordination. And I want to give you an example going back to what I talked about before, which is this example from 2D vertex models, because that's sort of how I started out my talk. So the L naught star, in this case, is P naught star. It's the special point where the transition happens, 3.81. And so now we're making the prediction that the sort of observed cell shape, basically in the rigid phase, what should first become rigid at the critical point. And then as I go deeper and deeper into the rigid phase, there's actually a small change in this cell shape that changes with fluctuations in the spring length and fluctuations in the area now. And so this is a plot that shows the observed spring length as a function, or the observed cell shape as a function of the target cell shape. Okay, and this is the transition point. So on this side, it's fluid-like, and this is just a plot showing you that the observed cell shape scales with the target cell shape. This is a line of slope 1. So this says observed shape scales with target shape. Okay, and on the solid side, I said, well, it's approximately 3.81, right? Because it is. It's basically stuck there, but that's not quite true. It's not quite stuck there, so you can see that there's a little bit of a slope here. And why that's important is because this is exactly the same physics that gives rise to the pitch in a guitar string. Very small changes in the guitar string give rise to very big changes in the behavior of the guitar string. And that's exactly what's going down here, so we'd like to understand it. Okay, and so this is now looking much closer. So this is a logarithmic scale on both sides. And now we're testing the hypothesis that this thing is linear in fluctuations in cell shape and fluctuations in cell area. And we find that it is. So what this is saying is that if I want to know about the mechanics of these sort of solids, these vertex model or biopolymer network solids, I can look at fluctuations in the cell shape and fluctuations in the cell area, and I can back out the macroscopic rheology of that system. Okay, so let me sort of explain what I mean. Okay, so all of these functions have parameters that describe sort of how the coefficients in front of these fluctuations. Okay, and we can look at all of these models. This is the spring network model. These are vertex models with various parameters. And what I'm showing you is that they all fit this model. The coefficients are different between models, but they're sort of robust to random realizations. So what I mean by that is our analytic result was just that this observed length or observed shape should scale linearly with fluctuations, right? But there's nothing that said that coefficient couldn't change from random realization to random realization. What I'm sort of showing you with data here is that actually those coefficients are pretty surprisingly robust to different random realizations, which means there's sort of a well-defined coefficient for a given model. Okay, so why is that important? It's important because once we write this down and once we know the coefficient, then we can make predictions about the bulk behavior, the macroscopic behavior, right? It gives us a way from going from a snapshot of a single cell shape or sort of a small set of single cell sort of spring lengths. And then it gives us a way to predict. So the last thing I need to tell you about is that this is sort of, if you think about it, changing L naught is changing the isotropic compression of my box, right? So those two are the same. I change my box isotropically. But I also expect things to change if I shear my box. And by symmetry arguments, you can show that that sort of coefficient, that should add a term that gives you something quadratic in the shear string. I'm not going to go through that argument, but that you expect something like this. Okay, and so then we can go and make predictions. I also wanted to highlight briefly that there's some really nice work by Mikhail Moshe, Mark Boeck, and Christina Marchetti, who's here, showing that something similar occurs in ordered 2D vertex model. This was just published earlier this year too. What they've basically done is really beautiful. They assume that they can use an elastic expression for the energy of deformation. So they forget about cell rearrangements. Okay, and so they use an elastic expression. For those of you who think about these things, what they're actually doing is looking at the difference between an observed metric, so what's the actual metric for a system, and a target metric for the system. And the sort of cool thing about this paper is that they sort of discuss that actually in these systems there's families of target metrics that preserve perimeters, and then there's a different family of target metrics that preserve areas, and then there's a geometric incompatibility between these two. And so this paper shows that for ordered vertex model lattices, you can actually compute these objects analytically as one parameter families, so you can do this minimization here really nicely. And this is the result of that sort of mixture of analytic and numerical work, that if you're looking at the energy, so this is the elastic energy generated by this calculation, by sort of changing either L naught or P naught, whatever you want to call it, with the cell shape, there's a critical point, L naught star, which in the ordered lattices is a hexagon. where this energy basically goes to zero, and then there's a system that has this pre-stresses due to geometric incompatibility, and then this green line is something that's floppy. Well, it's not exactly floppy, it's a very soft solid. Okay, so I just wanted to show you that actually there's some nice analytic calculations that can be done that can show that in ordered systems you can sort of think about this in terms of elastic metrics as well. Okay, so once we have all of this, what can we do with it? I've been promising it for a long time. Okay, so this is your observation, like what do you observe as the average spring length in your system? This is the length at the transition point, and these are these coefficients that you can measure by looking at fluctuations in the system or by looking at the response to shear strain. Okay. So the cool thing is that you can express the total energy of the system in terms of these control parameters, right, which is the isotropic compression or the spring rest length and the shear strain. And once you do that, then you can make predictions for you can write down the bulk modulus, you can write down the shear modulus exactly for these systems in terms of that energy functional, and then you can make predictions, analytic predictions about how changing the isotropic compression of your system or the shear strain in your system takes you from a floppy regime to a rigid regime. Okay, so what I'm telling you is this predicts the location of the fluid solid transition as a function of the parameters, the scaling relations. And so, for example, you predict that if you start in the floppy regime and you basically turn on a shear strain, then you find that the critical point where you become rigid scales quadratically with the shear strain with a coefficient phi, right, that sort of is a parameter of a given model. Okay, and so you can check that by basically looking what's the distance to the critical point, is and the critical shear strain. How far do I have to go out till I hit this line in numerics? And you find that yes, indeed, in fact, it's 2. Okay, but you can also do things like predict a jump in the shear modulus. So this is what happens. Basically, you say I'm floppy, I'm floppy, I'm floppy. Then I hit the rigid region, and then all of a sudden I sort of turn on a shear modulus, and you can calculate the magnitude of the shear modulus jump. You can say, okay, what if I if I compress a little bit and then shear, what do I see? And then you see this purple line, and you can say, well, what if I compress even more and shear the system? Then I see this blue line. Okay, and so why is that useful? It's because these things can be directly compared to experiments. You can actually, we haven't done so yet, but the idea is you could actually look at images of these fiber networks in these systems and see if the coefficients match up, right? And so this is just a picture now of the shear modulus as a function of shear strain, and you can basically see that as I change the compression, then I change sort of where I sit, and I go from something that looks like this blue curve to something that looks more like this purple curve, and then something that begins to look like steeper, like this red curve. Okay, one last thing I want to say that may be of interest to people here is that there's this big question in the literature on sort of extracellular matrix and fiber networks as to whether the nonlinear mechanics, so like I strain it and it's very floppy and then all of a sudden it turns on to be very stiff, whether that is generated by this geometric incompatibility that I've spent all of today talking about, or if it's generated by nonlinearities in the individual units. Because of course, biopolymers are themselves nonlinear, and so you might worry that that's actually the origin of the nonlinearity. And one of the really cool things that drops out of this scaling analysis is that there's a thing that you can look at, and I'm not going to go into all the details, but you can look at it in experiments as basically the shear modulus minus the jump in the shear modulus at the critical point, divided by the shear stress at the critical point and the critical strain at the critical point. And if you make this combination of numbers, this scaling theory predicts that there's a coefficient of 3. So it's not an exponent now, but it's a coefficient. And that if your system has that coefficient, then the onset of the shear stiffening is driven by geometric incompatibility. So it's a way of testing whether a system has a nonlinearity that's generated by a geometric incompatibility as opposed to a nonlinearity. So this is a really strong prediction, right? It predicts you plot this thing and it has a coefficient of 3, right, from the scaling theory, which is kind of cool. Okay, and the existing experimental data is consistent with that, but we should, it should be looked at more carefully now. Okay, so this is a list of the sort of analytical predictions that you can make with this scaling theory. And there's sort of been some observations of some of them in the literature, and then there's some, for example, these are just theoretical predictions and it's never been tested. So the idea I'm trying to get across here is that I could have had lots and lots of slides like this, but I won't bore you with them. But once you have a scaling theory like this, then you can make predictions about the behavior of the system in a wide variety of contexts that can be, confronted with experimental data. And the data that we have in epithelial layers so far, we haven't confronted it in full detail with this list, but it should be amenable to this as well. So both sort of collagen and vertex models or epithelial layers should be described by this type of thing. So it's very universal. Okay, and that's kind of interesting because, one of the things that you might be interested in is how cells, for example, in a tumor, interact with an extracellular matrix network. And what I'm telling you is both of those systems have a very similar rigidity transition in them. And so thinking about sort of either analytic or modeling or even predictions for experiments on that, it gives you a much nicer way of thinking about interactions between rigidity transitions as driving some of that behavior. Okay, I have about 5 minutes.
Yep.
Or maybe a little less, I have questions.
Wait.
But I'm standing between everyone and lunch, so it should be quick. Okay, so one thing, because there's been some mention of Darcy-Tungsten and also some mention of sort of dynamic processes in development. This is the cover page on growth and form, and it's a splash of milk. And later in this monograph, basically, there's these pictures of Hydra during development, and the suggestion is, well, maybe this is a lot like that. And I had a lot of trouble taking this seriously when I saw it because splashing is a dynamic process that requires a delicate interplay between thermodynamic forces, surface tensions of the kind Sev was talking about, or maybe you could call it tissue affinities or cohesion, right, something like that, and dynamic forces or viscosities, right? That's what gives you those patterns. And Hydra maintains its shape for a long time, maybe yours is a little too long, but a long time. So I was always worried about this. But I think the point is that if their system is really close to one of these rigidity transitions I'm talking about, then the relaxation timescales for the fluid can get very, very long. And if the relaxation timescales are long, the viscosities can be very high. So maybe it's possible, right, that this type of process could work. Okay, and so we've been studying Cooper's vesicle, which is the left-right organizer in zebrafish that sort of sets things up so, your heart, for example, ends up on the correct side of your body. And I'm not going to go through all of the details because there's a lot of them, but one of my collaborators has shown, so this is the organ. It's a little vesicle surrounded by like an epithelial layer, and the epithelial cells are monociliated, so they all stick cilia into the center, and then those cilia set up a fluid flow, and that fluid flow is read out to do left-right symmetry. The thing that I'm going to be focusing on is that my collaborator has shown that upstream of all of that, there's a specific set of cell shape changes in this organ that precedes left-right patterning. And so what Jeff has found is that if this is the anterior side, so sort of anterior side, and this is the posterior side, the cells on this side have to get really short and squat. And that means there's a lot less cilia on the bottom, right? And these cells on the top have to get really long and skinny, and that puts a lot more cilia on the top, okay, to make this thing function. And so since I'm interested in cell shape changes, I was like, well, maybe we'll try to figure out what's going on with these cell shape changes. And then we've worked on lots of different stuff, like besides what I'm showing you today, but when we saw these movies, we were really intrigued. So this is the Cooper's vesicle, and it's basically moving through the tail bud tissue. So those nuclei, the red cells are tail bud cells that are labeled, and this green thing is the Cooper's vesicle. And during the course of its development, it's moving through the tissue. Okay, so you look at it and you say, well, if there was a cylinder and I pushed it through a fluid, right, then I would expect that there would be sort of compressive forces at this side of this, and that there should be drag forces on this side of this object, and that would, maybe, if they were strong enough, elongate the cells in the back and compress the cells in the front and give rise to the shapes they see. Kind of crazy, but okay. So we can first do the simulation. This is like the very first check you might do. So this is just a 2D simulation of a vertex model where the cell shapes are perfectly, so there's no morphogen gradient that's generating an anterior-posterior asymmetry. All we're doing is taking a system that's identical in every way, except we're just driving it through surrounding tissue at a given speed. Okay, and so when we do that, you can see that the cells change shape in exactly the way that it looks like in the experiment. Okay, but it still has the problem that you wonder, okay, are the timescales right? Because, you know, this thing is moving fairly slowly, you know, in absolute terms through the tail bud tissue. And so there's some really beautiful experiments, again, out of Oche Campas's lab that have carefully measured the mechanics of the zebrafish tail bud. And let me see if I can get that movie is, there it's gonna play. So they take a droplet and they oscillate it. And so we can actually do the exact same thing in our vertex model. And so we can match up the time scales from the zebrafish tail bud tissue exactly to our vertex model. And then we can measure how fast in absolute terms this organ is moving through the tail bud tissue. And when we do all of that, you basically get, there's sort of this really sweet spot where the velocity, you know, to as best as we can tell, the velocities with which this tail bud is moving through, the KB is moving through the tail bud is exactly right to sort of generate the shape changes that we see in the experiment. Okay, and so now we're doing a more careful analysis, PIV analysis of the flows around this organ. And you see, so these are actually from data. These are sort of snapshots from data. And you can see that they're remarkably similar to the simulations and then also to an analytic calculation for what the flow field should be. And then you can sort of even start to move into 3D, to make 3D reconstructions of this thing. Okay, so people are interested in cell division, so say we're thinking about it, but there's still more to be done. Okay, so biological tissues exhibit rigidity transitions that are important for biological function, and the point, one of the most important points I think is that successful models for these systems are under constraint. Like if you did constraint counting, you would say they're never going to be rigid. And so in order to get rigidity in them, you really need this type of geometric incompatibility where sort of your box size doesn't match up with the inherent sort of properties of the objects. Okay, and the rigidity in these models is generated by an onset of residual stresses. And then the critical new thing, I think, is that there's a minimal length function, we think, that describes all of this behavior, and that gives rise to a bunch of scaling laws that make sort of unifying predictions about a lot of experiments in both acellular and cellularized tissues. Large relaxation timescales that occur near these rigidity transitions can perhaps be harnessed to generate long-lived organ shapes via drag forces, and there's a lot more to do. Thank you.
I have a question about the network, or a question about the polymer networks.
Yes.
If the network were put together in such a way that the junctions could slide, they weren't fixed, does that change anything?
Yeah, so you're saying, like, what if the crosslinkers weren't the solid kind, but were sort of these movable kind? And so, yes, it would change. So we actually are doing some simulations now with 2 additional ingredients to this model. One is I sort of I put a big X over bending rigidities, and so different crosslinkers generate different bending rigidities to the system, and that will smooth out some of the discontinuities that I showed in the model. So that's probably the 0th order change that crosslinkers will give. And then mobile crosslinkers, I think that they'll still give you sort of that, the sort of same constraint counting, but they'll probably, I would guess that they would introduce another time scale that allows, depending on how long they take to move relative to the sort of other time scales of the problem, they could introduce a fluidity on that time scale. But we haven't done it.
Have you thought of including the effect of the fluid in between the polymer network, or in the case when the swelling happens?
Yeah, so you're saying, did we account at all for sort of hydrodynamic effects for the fluid in between the polymer network? No, this is a dry, sort of, the movies I showed you are for sort of simplest spring network with no hydrodynamics. and even no bending. So there's a lot more additional things that can be added to the model. One of the things I'll say is that other researchers have shown that adding those things to the models tends not to change the overall, because people have sort of studied these things numerically and theoretically before, that they don't change sort of the scaling relations that you see, but they do modify sort of some of the discontinuities and they smooth out some of the behavior.
So I'm wondering about dimensionality. So you've talked about two-dimensional systems and one-dimensional systems, but I guess if you're looking at kind of more constrained systems or thinking about pseudo-stratified tissue layers, right? So the dimensions are not necessarily very clear, right, in that sense. So maybe you could comment on that.
Yeah, so I think one of the things, one of our first clues that that these systems couldn't be like particulate jamming is that, I told you that in sort of particle-based jamming, the degrees of, that the dimensionality of the system plays a really important role in when the system rigidifies, right? The number of constraints I need is proportional to the dimension. And so we actually studied 3D Voronoi models, and we're actually working with a couple of, I showed you those pictures of those breast cancer sort of cell culture aggregates. We actually have 3D segmentation data on those. We can compare it to 3D versions of these models. And all of this seems to go through in 3 dimensions. Now, there's an additional thing which is pseudo-stratified, right? And if you have sort of, let me call it pneumatic order, if you have ordering in the system, then that adds an additional ingredient which you need to account for in these sort of rigidity models. So you can think of it sort of as in sort of pneumatic ordering, which is like if I have cells and they're sort of elongated and then that direction of elongation is aligned, then I need to sort of account for that alignment. And you can think about that alignment as an additional axis on some phase diagram. And you can actually we do have, we've developed a theory that sort of says how the transition line between fluid and solid changes as a function of ordering as well. So the short answer is 3D works very much like 2D for these types of models, in contrast to other types of models. So it's kind of cool, I think, that once you have this type of under-constrained system, you can go from being a monolayer to being a pseudo-stratified layer to a 3D without really changing your organizing principles very much.